इसको
तो
भी
अभी
मैं
हम
बंद कर
लोग
देता हूँ फिर।
ब्रॉडकास्ट, हाँ सर अभी बंद कीजिए, सोमा आपको बताएगी कि
श्योर श्योर।
आपका वो इंट्रो देने के बाद आपका इंट्रोडक्शन
श्योर श्योर श्योर।
करवाएगी, फिर हम लोग स्टार्ट करेंगे।
श्योर।
तो अभी हम लोग जा रहे हैं यूट्यूब। हाँ, हम लोग ब्रॉडकास्ट शुरू कर चुके होंगे अभी। 54 वेटिंग दिखा रहे हैं। जस्ट मैं एक बार कंफर्म कर लेता हूँ कि
ये
हम लोग
लाइव।
ओके, वी आर लाइव। Thanks everyone for joining our today's webinar. It is our 66th webinar in the series, and we are very happy. Today we got a young, uh, energetic and dynamic scientist from Nabii. We are met together before one and two times, but this is our opportunity to invite him for this Bioingene webinar, and we are very excited about. We all know about the golden rice, but now we can learn something about the golden banana, I hope. And today's topic is very important, as you know, is the one side of a coin, like food security, and another is nutritional security. So it is a very important topic, and we are trying to cover in bioengine food security and nutrition security, and we are invited renowned scientists all over the world. So I today Shoma will help me to broadcast these sessions, to moderate it. And now Soma will go for the introductions to the speaker, then the talk. Then in the meantime, we collect the YouTube live questions. Ultimately, there is a questions answer round, and finally, some question we will also ask from the live audience. And don't bother about the link for certificate application. It will be provided at the end. Those are already registered for this webinar and able to submit their feedback. They will able to get your certificate after 4 or 5 days from bioengine.com website. So, Shoma, please start the webinar.
Yes, Shubho, please start the recording.
Okay. Okay. But start. I'll start.
All right. Hello and welcome to Bioengine webinars. Bioengine is a non-profit organization created to promote plant research worldwide. This is a platform from which plant scientists can present their research to the world, and we hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on building the future of plant sciences. We are thankful to our speakers and the viewers of Bioengine family for your interest in the Bioengine webinar series and for your appreciation and of our efforts. You can register for Bioengine webinars through our website bioengine.com or our social media pages. You are live viewing this webinar and you can watch it again later on the Bioengine YouTube channel. So please remember to subscribe so that you don't miss out on any of our. After attending today's talk, you can apply for a certificate of participation via the feedback link which will be provided in the YouTube chat after the presentation. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is crop biofortification for nutritional security. Our guest speaker today is Dr. Siddharth Tiwari, Scientist F, National Agri-Food Biotechnology Institute, Mohali, Punjab, India. His area of specialization includes plant tissue culture, molecular biology, and genetic engineering. His group is presently working on the metabolic engineering of staple crops like banana and wheat for nutritional enrichment by using modern biotechnological approaches such as transgenics and CRISPR-Cas based genome editing. He developed international research collaborations and worked together under various intergovernmental schemes with Australia, USA, Germany, Poland, and Thailand. His work is much talked about, and especially after his interview in 2018 in the Rajya Sabha National Channel and in 2022, the program telecasted by Vigyan Prasar, Government of India. His banana genome editing work was celebrated as the top 15 promising innovations of Indian sciences in 2018. He has been elected Fellow of the prestigious Plant Tissue Culture Association of India. His doctoral research earned him many awards, such as the BioAsia Innovation Young Scientist Award 2010 and the Indian Science Congress Association Young Scientist Award 2010. 7 students were awarded PhD under his supervision. He has filed 4 patents and published 3 edited books and several book chapters. He has trained more than 80 graduates and postgraduate trainees in various plant biotechnology fields and also delivered more than 100 lectures at different platforms for creating awareness about plant biotechnology applications and about his research work. His research findings have been published in more than 50 peer-reviewed journals of high impact factors like the Journal of Experimental Botany, Metabolic Engineering, Functional and Integrative Genomics, Plant Science, the Plant Journal, the Biotechnology Advances, Frontiers in Plant Sciences, Genomics, etc. We are highly honored to have him with us today. And I would now request sir to please, we can start today's talk and you can share your screen now.
Thank you so much, Dr. Soma. I am really thankful to Dr. Majumdar and you for giving me an opportunity. Just briefly, I will introduce about my research work as well as my thoughts about biotechnology, especially in the case of their applications for crop improvement like biofortification. So now I am going to just connect with my slides so that please let me know if it is visible.
Yes, it's coming now. Yes, visible.
Now in that presentable mode.
Yes.
Yeah. So thank you so much, and I really welcome to all the audience. Those are connecting with these presentations, and as Dr. Soma have already been mentioned that I am working as a scientist at National Agri-Food Biotechnology Institute. This institute basically comes under the Department of Biotechnology, Government of India, and located in Mohali, Punjab. So these are my email ID as well as my the Twitter account. If somebody has any kind of interactions in future, also they can directly contact me. So going to details about my presentation, first I would like to let you know about the foundation of my research, where it is started and where it is going. So I think most of you already know about DNA because I know that there are some students, school students, as well as undergraduate students are also the part of this webinar. So I would like to briefly introduce about that foundation of my research, and it is started by DNA. As you know, DNA is basically a genetic material, and it denotes for deoxyribonucleic acid, and it's a very small. You can't see by naked eye. You have to go to see in the microscope, and when you will see, it's in the double helical structure. That is in the left side that you can see. Wait, I will pointer. Here you can see this double helical structures, and it is bound by four bases, also known as the nucleotides: adenine, thymine, guanine, and cytosine. So second part of my that foundation of my research is plant tissue culture. That plant tissue culture is basically a techniques where you can be use any part of plants for tissue culture work, and you can be multiplied to generate several genetically identical plants. So these two are the basic fundamentals of my research. One is the hereditary material or genetic material known as a DNA that I have used as a improvement of traits. How it works, that I will let you know in my few slides. Then plant tissue culture is the techniques that I am implementing as a basic part of this to develop a genetically modified crop. So you know that all living organisms, including microbes, humans, plants, or animals, they have one common things that is known as a genetic material or nucleic acids, the DNA and RNA. And that genetic material DNA is basically heredity, and it is transferred from one generation to another generation with certain traits. And it is also playing major role for the evolution from the for the all prokaryotes like a lower organism, microbes, as well as higher organisms like eukaryotes. So here you can see that each and every cell of all living organism has contained genetic material that is known as a chromosome, and this chromosomes basically have genes or DNA, and this gene or DNA is going to be transcribed into the form of RNA, and it RNA is translated into the form the end product known as a protein. So phenotypically, whatever observations you are seeing, or just you can visualize like a color of eyes or skin color or height or plant's leaf size, all that phenotypic characteristics that is governed by the DNA that is translated into the form of protein. So if there is any changes happening in this genetic material or DNA, it is having a huge impact on a particular organism. I think most of you know that in human population, when you will see that whatever that changes or differences we are observing, it's just due to the changes or this mutation in 0.1% of the whole genome. So 99.9% of the genome of all humans are same. Only 0.1% changes has been shown the variation in our populations. Like you can see in the different colors of the skins, height, eye color. There are so many changes that you can see, and those have been governed only from 0.1% mutations or changes. So how much impact of mutation you can see over here, and if there's any changes that intentionally or unintentionally we can do in the genome of any living organism, it will have huge impact. So this is the base of my study, where I am trying to utilize that genetic material. Those are carrying important characteristics or features that we would like to utilize for the improvement of traits in the plant crops. That I will let you know how I am utilizing this genetic material or DNA in our research. So you know that about the central dogma concept that I have already been explained. This is the double helical structure of DNA, and it is going to be transcribed to form the RNA. And this RNA is basically contain the all information that is going to be converted or translated into the form of protein. And this protein is basically provides some features or phenotypic observations as I have already mentioned in my previous slides. But you can see that this particular concept is known as a central dogma. You have already been read about this in your syllabus book, especially for school students or college going students. They understand the concept of central dogma, that is from DNA to RNA and RNA to protein. So protein is the end product after translation that has been governed the particular traits in the living organism. But in some of the viruses, most of the viruses, basically they contain RNA as a genetic material, and they are going to be reverse transcription. That means this RNA is going to be converted into the DNA, and that is known or governed by that one of the enzyme known as a reverse transcriptase. So that is only happening in the viruses, not for other living organisms. So now about plant tissue culture. Plant tissue culture, that one concept I think most of you know about totipotency. That means each and every plant cell has capacity to generate the whole plants. And this totipotency is the basis of the plant tissue culture. How it is? You can see over here, that is a plant. In this plant, we have taken the one cell that is from the leaf tissues. This one cell, single cell, is containing the different organelles, including the chromosomes in the nucleus. And this chromosome has containing the genetic material. And when we are going to be use this particular cells in a particular environmental conditions, and it is converted into the whole big plants. That is the capacity of plants and known as a totipotency. But in case of human, it is not possible because human skin cell is able to form the skin cells only, but not to the nerve cells or other organelle cells. So this is capacity is happening only in the plants, and that is due to the plasticity, and it considered as a totipotency concept. So you can see over here, like this example is clearly showing this is the plant, particular leaf portions. It is known as an explants have been taken as a source of explants or one part of the plants, and it is going to be planted or just transferred into the artificial media that is known as a growth media under the in vitro condition or sterile environmental conditions. This explants or plant tissue, we are just first sterilized, and after sterilizations we have gone through the putting on that particular media combinations or under that artificial nutrient conditions. And this particular cell or tissue is going to be multiplied and form the callus. Sometimes it can generate direct plants, and it is going to be sometimes multiplied. And after multiplication, this cells is able to generate the whole plants. So that all things are happening under the aseptic conditions and providing artificial environment as well as artificial nutrient conditions. So you can make the control to generate whole and multiple identical plants. Those are resemble to parents because the genetic material is same. So this is the concept of plant tissue culture that I am going to be implement in my research. And you can see that it has so many applications of plant tissue culture. Like you can rapidly multiply large number of genetically identical plants because it is containing the same genetic information. Those are happening in the their parents tissues or parents plants. So the all subsequent generation are carrying the same genetic material, and it is has so much impact on the plant tissue culture industries, especially for banana and many ornamentals and fruit crops. Those are commercially multiplying by using plant tissue culture. The second is it requires that less space and fewer resources as compared to the traditional methods where we have to be use seeds and that can be used for that multiplication and subsequent regeneration. But in case of plant tissue culture, you can take any living parts of the plants and that parts you can go under that certain environmental conditions as well as low space in the laboratory conditions only. You can be generate a huge number of plants, million of plants, and those can be grown in that tissue culture laboratory. And if required, then you can transfer those plants in the field conditions. And it is a round year production. It is not dependent on the environmental or seasonal conditions. You can multiply all that plants depends upon their what we can say requirements or market value. Then you can also preserve germplasm. Those are required for genetic diversity or your research purposes. Those particular germplasm you can be just preserved under that in vitro laboratory conditions. Then so many plants or so many species, those are going to be considered as endangered. That means those plants or those species have very limited numbers and it is considered like endangered. So those plant species can also be multiplied and preserved for a long time, and if required, that can be utilized for growing in environmental conditions. And next is the genetic modification. That is basically related to my this particular presentations. By using plant tissue culture as a tool, what you can do, you can create the abiotic stress resistance variety where like different abiotic stress like salinity, drought, heat, those are directly linked with climate change conditions can be tackled by using genetic modification. You can also develop the tolerance variety against to biotic stresses like various diseases as well as insect pests usually attack to the crop plants. You can introduce the new traits or new gene that has been given the protection against to these stresses conditions, and also you can be enhance the nutrition value of a particular crops, that is known as a biofortification. And that biofortification, we have to be identified a gene, those might be responsible for the enhance a particular nutritional traits. That how I did in my experiments or my research, that I will let you know in my few slides. But before going to in details, we have to understand about genetic modification, how we can do this genetic modification in crop plants. So there are different approaches. Some of you already may know about these approaches like conventional plant breeding. In this case, what happened? You have to have one variety. Those are known for the disease resistance of particular traits. This is just example for particular characteristic known as a disease resistant. But similarly, in other cases, like some varieties might have an nutritional enriched or some other traits specific. But those variety are not commonly growing because those are having low yield, and they have what we can say not that sufficient acceptance by the farmers, but those have been characteristic showing the resistance of particular traits. So what happened that other cultivated varieties, those are usually susceptible to that particular traits. You have to go to cross, because these two varieties are related to each other, but one variety is having that particular traits, but other variety is susceptible to that particular traits. When you are cross by using that plant breeding methods, then what happened that particular traits of this variety that is known as a resistance or improved traits is going to be transferred to your allied variety, that is a basically susceptible variety. After subsequent generation, you will see that after crossing these two varieties, what happened, the trait has been transferred from this variety to the allied variety, and subsequently your cultivated varieties is converted into the resistance or improved varieties. But it takes long time and it is linked only with the related varieties. Like you can go to cross between wheat to wheat, rice to rice, but not wheat to rice or rice to banana. So this is that disadvantage of this technology. You can make the crossing between the related varieties only, and it requires a long time to introduce that traits. And also some of the undesirable traits can also be transferred with your desirable traits to this allied variety. So these are certain limitations of conventional plant breeding. Second one is known as a mutation breeding. And what happened in this case, you have to use some chemical mutagenesis like EMS or radiation mutagenesis like gamma radiations. And you have to be incubate your seeds or plants by this chemical or radiation mutagenesis, and certain mutations or certain changes will happen in that your particular crop varieties or crop plants. That mutation you have to be go to screen, because you have to be generate a large number of populations, and after screening you will identify okay, there is a one mutation that is desirable for you and it is providing certain traits like disease resistance, nutrition quality, or like that. So that particular variety or what we can say seeds you will select out for the subsequent generation and you will be analyzed. But again, because you are putting that random chemical or radiation mutations, so there are so many changes happened in that particular crop's genome, because this is a randomly happening in the genome, and identifying that desirable crop plants with certain traits is very, very difficult and challenging task. And it is a very laborious, you have to generate several plants and several generations to see the stability of traits. So this is also a limitations of conventional nutrition breeding. Now, third one is the plant genetic breeding, or also known as a transgenic approach or genetic engineering or recombinant technology. In this case, what you can do, you can take the gene from any living organism that has been carrying important traits that you would like to introduce in your desirable plant crops. So what happened, that gene you can take and clone in the plasmid, and that plasmid is going to be performed by tissue culture transformation methods in that particular crop plants, and Then what happened? This gene is going to be integrated in the genome of this particular variety. Those are usually susceptible to the disease or deficient with the nutrition. And that traits or specific traits is going to be introduced in the genome of that particular variety. And it is considered like transgenic because you have taken gene from other living organism. So this method has also one drawback. It is a random integration in the genome. So that gene, whatever you are going to be introduced in that variety, it is randomly integrated in the genome of that particular plants. So there are sometimes what happened that gene is going to be disrupt the function of other important traits. So you will get sometimes off target kind of effects. So how you can just reduce those kind of possibility? Then you have to be generate several transgenic events, and subsequently you have to be analyzed and identified particular plants. Those are having less, what we can say, negative impacts or no negative impacts of that introduction of a particular gene. Based on that, you will identify it and go for that their environmental trials, open conditions, as well as subsequently release for the commercial cultivations. The most recent and advanced technology known as a genome editing, that is also known as a new breeding tool. It works very precisely. How it is precise? Because if you would like to target a certain genes, those are not required in that particular crop plants. Like that gene is responsible for the anti-nutritional factors, or that gene is responsible for the providing susceptibility to the disease, or it is responsible for the depletion of nutrition. What happened? That gene you can be silenced by using or create mutations on that gene by using genome editing approach. What happened? That gene will not be functional, so that desirable undesirable traits will be shut down, and you will see the desirable impact in that particular crops. And important is that this is working in a very precise manner, so that other gene will not be disturbed. This is the advantage of genome editing. Even you can introduce the new traits in a precisely in the genome by using genome editing. How it will work, that I will let you know in my subsequent few slides. So my whole presentation is basically focused on two approaches. One is transgenic and second one is the genome editing approach, where I would like to use these two approaches for the improvement of certain traits in crop plants. So that is my PhD work. Here I have developed insect resistance variety of groundnut. What I did, I have selected one gene known as a Cry1 EC gene. I think you heard about BT cotton. And in BT cotton, there is a Cry1 AC gene has been considered to develop resistance against certain insect pests. Similarly, in my laboratory during my PhD at NBRI Lucknow, what we did, we have designed Cry1 EC gene, and this gene is particularly effective against one insect known as a Spodoptera litora or tobacco cutworm. And this insect is basically attacked to the groundnut leaf, that peanut leaf or Mohali leaf, and completely destroy the crop. In southern part of India, like Karnataka, it has been reported nearly 70% of the yield loss due to this particular insect. So what I did, I have used that groundnut seed derived this explants known as a cotyledon. I removed this embryonic portions, and this cotyledon I cut in that vertically and developed this de-embryonated cotyledon explants. And this explants I have used as a source of the tissue culture for multiplication or generation of multiple plants. In this particular media combinations, you can see several shoots have been generated from this particular explants. And these shoots have been transferred into the rooting media, and after rooting, it is acclimatized in the soil pots and subsequently transferred into the environmental conditions or in the greenhouse. So you can see what I did during this tissue culture process. I have designed this Cry1 EC gene that has been known to provide protection against to this insect, Spodoptera litora or tobacco cutworm. I introduced this gene in the plants of groundnut by using tissue culture, one of the tools. And after introduction and identification of that gene in that crop plant, this new gene, there are certain methods like PCR, southern blotting. I make sure that the gene has been introduced in this native plant crops, And then I have checked their traits because this gene is responsible to make some product known as a BT or cry protein. And this cry protein has capability to die. Insect is going to be die when it chew this cry protein. So my plants, those have been developed by using cry1 EC gene expression. It when we had performed insect bioassays, you can see that different stages of this insect are not able to survive when they chew this transgenic leaves where cry1 EC protein is expressing. While in case of non-transgenic leaves or control plants or natural plants, you can see that insects are not able to survive. Because insects are able to survive and they easily grow as well as they can completely chew or completely finish this leaf. So this is the laboratory based assay. It makes sure that insect is not able to survive when we are putting those in that transgenic leaves. So this work is I have used to targeted this protein only when insect is going to attack and where the promoter is playing major role for the expression of gene. And in this case, I have used one wound inducible promoter. So our protein or our gene is expressed only when that insect is going to be attacked. And rest of the times, this protein will not be produced. This is the beauty of this experiments. And you can see several publications have been published during my PhD from NBRI Lucknow. And due to this work, I got Indian Science Congress Young Scientist Award in 2010 in the section of plant sciences. And you can see that our honorable president as well as the Bharat Ratna, Dr. Abul Kalam Azad has been given me this award and I have brief interactions and discussions about my future as well as my work in this direction. Then my second objective of PhD is to use plants as for the production of vaccine. I think some of you know that plants can be used to express vaccine candidate also in a targeted manners. Similarly, I did in case of groundnut, and it has so many advantages because you can easily multiply. We know that plants can be easily multiply that environmental conditions, and it is a low production cost as compared to the other cell line based or human based assays that human or animal cell lines usually used for the production of vaccine, but it is laborious and very costly and easy to contaminate. But when plants can be used for the same purposes, it is easily multiplied and can be a what we can say produce in the large number, large amount. And when specifically if you would like to produce this kind of vaccine in the seed, it has several other advantages like seed content a high amount of protein. It can be easily you can get even uncooked condition. There is no need to go for cooking, especially groundnut seeds or peanut seeds. And it content low water so that you can be store those particular seeds for long time at ambient temperature. There's no requirements of refrigerations. So those are certain advantages if you are using plants as a source for the vaccine production. What I did, I used this groundnut seed to express vaccine against to rabies as well as cholera. I have designed that rabies glycoprotein, that is a spike protein, at their fusion with cholera toxin B subunit, and that particular antigenic combinations in the form of gene and that construct we have transferred by using tissue culture approach in a targeted manners so that that particular vaccine candidates expressed into the groundnut seeds only. And in this case, I have again used another promoter that is known as a legumin promoter. So that vaccine is going to be expressed in the seeds only. And you can see that this is one of the estimation that we had published in the Journal of Biotechnology Advances. And we have mentioned that if we can produce that vaccine candidates in the groundnut seeds, and that expression is 1% of the total protein, so that from 1 acre of the groundnut crops, you are able to produce vaccines that can be provide protection against to 468 million people. So this is the beauty, and you can say what we can say advancement or advantages of using plants as a source for the vaccine productions. And very recently, I think some of you heard that Canada is the first country in the world that have been used plants for the vaccine production against to COVID-19. And that vaccine was commercially utilized for their populations and it was released in 2020-22. for the vaccinations of adult as well as old people in Canada. So it is providing efficacy and as well as protection against to this virus. And that vaccine has been produced in the leaf of tobacco plants and where it was purified and used for immunization of the populations. So this is that one of the very famous examples where plants can be used for the production of vaccine, and it will be a very reliable and very efficient source in the future when this kind of pandemics happen in the world, so that we can be use plants for the production of vaccines. And for my work on the use of groundnut seeds as a production of vaccine against to rabies and cholera, I got another Young Scientist Award that is known as a BioAsia Innovation Award in 2010. So subsequently in 2010 February, I joined NABI, that is known as a National Agri-Food Biotechnology Institute. And this institute is located in Mohali, Punjab, as I already mentioned. And our institute vision is very clear: to provide food and nutrition security through agri-food biotechnology research and innovations. So you can see that following our visions, I am working on the biofortification and where I am utilizing biotechnological approaches like gain of function, that is known as the overexpression. So that I mentioned that we can develop transgenics or cisgenic crops where that gene can be overexpressed, and we can also disrupt the function of genes known as a gene silencing or using CRISPR approach. So I am utilizing these all approaches in my research where I would like to introduce or improve the crop plants for a certain traits. So in this presentation, what I am going to present, I will let you know about genetic engineering or transgenics or cisgenic or genome editing approach, how this approach basically work, how you can be implement, what is the regulations of this approach, and how it is implementing for the crop biofortifications. My second part of this talk is basically covered my own work where I have engineered banana for the improvement of nutrition, and that nutrition is basically for provitamin A or precursor of vitamin A, that is known as a beta carotene. So I will try to cover all these two particular topics in my presentation, and after this I am expecting that participants will be able to get awareness with the recent plant biotechnological techniques and their regulations. They can able to raise genetic engineering plants with novel traits like agronomically improved crops, nutritionally improved crops, or climate resilience crops. It will also help in writing a project proposal in the field of plant biotechnology. This is my expectation of this presentations. So you know that by what is that plant biofortification? I think you know that most of the nutrients we usually get from the plants, and those nutrients are like provitamin A, iron, zinc, then calcium. There are so many nutrients. Those are known as the micronutrients we can usually get from the plants when we eat those plants or their products. And if these nutrients are less or what we can say minimum in that amounts in that particular crops, then we will have the deficiency of that particular nutrition. And that is considered a malnutrition or micronutrient deficiency or hidden hunger. So this is the problems. If that plants are deficient to particular nutritions, then we will not be able to get that nutritions, and it is considered like a hidden hunger or micronutrient deficiency. So how you can get these micronutrients or how you can be rid of this malnutrition problem? There are three major ways. One is the nutrient supplementation, where you can include that nutrition in your diets. Additionally, exogenously, we can say. Then dietary modification, you have to have that diverse dietary source. And third one is the biofortification. When you will talk about nutrient supplementation and dietary modification, it's a costly approach. Poor mans are not able to purchase these kind of costly diets, and they will not be able to go for diversification of their diets, because most of the poor populations are dependent on the staple crops like wheat, rice, maize, or some fruits like banana, because they are not able to purchase those costly what we can dietary sources. Then biofortification comes in the picture. Biofortification means you can use the approach where you can improve the nutritional quality of crop plants. So this you can do by three ways. One is the traditional plant breeding approach. There you know that one traits is available in the one particular variety of this crop, and you can go to transfer that that trait into the allied varieties by breeding or crossing approach. But as I mentioned, it is limited by the limited that related crops only. You can't go to cross that wheat to rice. You can go to cross only wheat to wheat or rice to rice. If that trait is not available in the any variety of rice, you will not be able to improve that rice variety. Those are deficient with certain nutrients. So this is the limitation of traditional breeding. Second one is the agronomic approach. In this case, you will go to spray the particular nutrients in the crop plants at a certain stages of plants, like fruiting stage or that is their vegetative stage. So what happens? This nutrients will be absorbed by the plants and it will be relocalized in a particular tissues. But again, it is a basically not a one-time investment. Each and every time you have to be spray those particular plants, and then you will get that particular nutrition. So it will be considered like a costly, and many times you have to be and what we can say implement this approach. Then third one is the genetic engineering. This is a one-time solution basically, and it can be go to transfer traits from any living organism. Even you can take the gene from microbes and also from the big animals like whale or like elephants. So this is the beauty of genetic engineering, where you can get that particular genes, those known for that particular traits, go to transfer that susceptible or allied varieties. And also genetic engineering, as I mentioned, you can be disrupt the function of genes by CRISPR approach. That gene is not required for plant crops. So these are the ways that you can be used for the improvement of nutrition quality in crop plants. But we are going to be focused mostly about genetic engineering approach. How you will develop genetic engineered plants? There are several ways, and here you can see that there are certain steps are involved for that genetic engineering. As I already mentioned, plant tissue culture is the base for this. And here you can see the regeneration or multiplication through plant tissue culture is very, very important. That protocol has to be optimized. And then you have to be identified the gene that you would like to go in for transformation, or would like to introduce in the crop plants, or you would like to be silenced by using CRISPR approach. So you have to be identified that particular genes that you would like to introduce. Then you have to go for again molecular biology or genetic engineering approach, where you have to be perform transformation, and that transformed plants have been regenerated again by tissue culture approach. And after regeneration, you have to be but we can say go for screening using different molecular biology techniques like PCR and southern blotting. And after confirmation, you have to have perform agronomical and biochemical analysis to see that your traits are introduced or not. So these are the stages. But when you will see these particular stages like selection of target genes and after generation of plants to analyze that bioinformatics are playing major role specifically for the genome editing. How it is playing important roles that I will let you know, because there are softwares those are playing to predict the function of that gene, and that gene you can be utilized for designing of guide RNA and by then subsequently implement by using genome editing approach. And after that plants edited lines have been generated, you can be analyzed again by using bioinformatic tools, there is a changes in the genome or not. So that changes like there is any kind of mutations are happened or not, that you can be again analyzed by using bioinformatic tools. So these are that some of the stages that you have to be know very well about implementing any genetic engineering approach in crop plants. So I have nearly more than 20 years experience in all these genetic engineering approaches, and I am also working on CRISPR approach when I joined Nabii. So for plant genetic engineering, specifically for biofortification, why it is needed, why it is required. There are three questions come in our mind. First one is the population, second one is the hunger, and third one is about the hidden hunger. The population, you can see that this is a global population status. And today I have taken this information from the internet by using this particular website. You can see that world population has already been reached more than 8 billions. So there is a more than 8 Arab people in the world, and India is the highest populated countries, and it is accounts around 1.439 billion people. And after that, China. Earlier, China was the highest populated countries before 2 years, but now India is the highest populated country. You can see that, and this is that prediction it was predicted earlier that India will be highest populated countries in 2025, but this target India has achieved before 2 years. 2023, we were the highest populated countries. You will amazing that how much food requires for this population. And this is the hunger index has been published very recently in 2023 by Global Hunger Index. Here you can see that they have considered 125 countries in the world to see that which country is most hungered. And in 125 countries, India position is 111. So we are in the positions like this, and our other neighboring countries like Pakistan, Bangladesh, Nepal, Bhutan, many South Asian countries as well as Southeast Asian countries and African countries are considered serious conditions of hunger. So most of the people of these countries are not getting sufficient food for their daily requirements. So across world, it is estimated nearly 828 million people go without food every day, and they are considered hunger or undernourished because they are not getting sufficient food. So India accounts for highest undernourished people in any single country. That was the reports published by UN in 2023. So scenario is not very good, and most of the children as well as the women are suffering with several nutrient deficiency also. So you can see that most of the populations are not able to get sufficient food in their diets because India is the largest populated countries in the world. So how we have to be provide sufficient food of these populations? That is the question number one in our mind. Second one is the hidden hunger, or third one is the hidden hunger. As you know that hidden hunger means micronutrient deficiency. This is not the India problem, but it's a global problem. You can see around more than 2 billion pupils. Is out of 8 billions, 2 billion pupils are worldwide suffering with hidden hunger. And these micronutrients are mostly from vitamin A, vitamin C, iron, zinc, iodine, and folate. So these micronutrient deficiency are the major problems in the world, and it is reported around 7 million children under the age of 5 years die per year because they are not getting sufficient micronutrients in their diets. And this micronutrient deficiency is responsible for many abnormalities like impaired brain development, stunting growth, mortality, impaired immune systems that leads to the risk of various diseases like diarrhea, malaria, and measles. So you can see how you define that micronutrients. Micronutrients are the small fraction of our balanced diet and plays essential roles in human metabolic process. So if deficiency of these micronutrients lead to the several diseases as well as mortality. And mostly children and pregnant women are suffered by this deficiency. So how can be rid of this micronutrient deficiency? You can go for diverse dietary sources, but as I mentioned, most of the populations are not able to purchase these costly, what we can say, food sources. So they are mostly dependent on the staple crops. So our target to enhance these particular nutritions in the staple crop plants by using genetic engineering approach. Now we will see another facts about India. You can see that Indian population has been reached more than 1.4 billions. And when we consider that one out of the every six people in the world is basically belongs to India. And when you will see the globe, there is a 71% is belongs to the water and 29% is the land only. And in this 29% of the land, India belongs to 2.6% of the land. And in this 2.3%, sorry, 2.3% lands are utilizing for the production of crop plants for our dietary sources, living expenses or urbanizations, globalizations, there are so many things that we have to do in this land that is belongs to India. It's at 2.3%. And you know that nearly 60% of the Indian populations depends on the agriculture. So how much food we have to be produced by this particular lands? That is another question because population is increasing day by day. So it is expected in after 5 years or 6 years, what will happen? The population have already been reached more than 8 billion, so that it requires so much food, and it is expected that undernourished people or hungry people or those people are not getting sufficient food, daily requirements is more than 1 billion. So that nearly 1 Arab people out of 8 billions are not able to get sufficient food in their diets. And it will increase the pressure to protect the environment, biodiversity, and conserve our natural resources. So how we resolve this kind of issues? So then we can go for plant breeding. As I mentioned, we can improve that crop plants, but it has limitations because you can go to cross in the related varieties, and that variation should be available in the germplasm only. If there is no variation or that particular traits is not available in the related varieties, you will not be able to perform breeding. Then how it will be rid of these problems? Then you will create the additional variability, and any additional variability can be created, or we can influence the desirable genes from related or unrelated species by using modern genetic engineering approaches like transgenics or genome editing. So these approaches come in the pictures where we can be improve the traits for our population's requirements. So transgenics or cisgenics, I will let you know about basics, regulations, and their applications. You know this terminology like genetic modification, genetic engineering, recombinant technology. These are the synonyms to each other. So transgenics or GMOs are defined where that gene of interest can be introduced by using recombinant DNA techniques. So the bottleneck of that plant breeding as well as genetic engineering approaches are years. Like as I mentioned, that conventional breeding required that linkage of undesirable traits. It's a long time or time consuming process, and it is performed only the related varieties. But similarly, in case of genetic engineering, you need to optimize the protocol of regeneration and genetic transformation. You have to know the function of that gene, and you have to be develop good laboratory practices and follow the biosafety regulations. So these are that some bottlenecks of genetic engineering as well as conventional breeding. And plus or positive points about genetic engineering is that you can be perform or transformation or can be transfer the gene even across the taxa. There is no limitations. Any living organism gene that is known for a particular trait that you would like to get, you can go for transformation or genetic engineering. So how you will perform genetic engineering? There are several methods are available, like biological methods like agrobacterium mediated transformation, implanter methods, fluoridip methods, then physical methods like particle bombardment or biolistics, and chemical methods like PEG mediated transformations. So, but most of the two common methods like biological methods through agrobacterium mediated transformations or physical methods like biolistics are very commonly utilized in the laboratory to introduce the gene in a plants. How it will work? Here you can see that agrobacterium mediated transformation. This is one of the examples. In the natural conditions, what happened? This bacteria is containing this particular genetic material, and this genetic material is known as a TI plasmid. In this TI plasmid, there is a tDNA, and this tDNA is containing some tumor-inducing genes. So when naturally this bacteria is infected to the plants, this transfer this tDNA into the plant genome, and it is going to be integrated in the plant genome, and subsequently you can see that whole plants have been generated, but this gene is already introduced in the genome of particular plants. It is going to be synthesized some opines, and bacteria likes those opines or metabolites for their nutrient requirements. So these genes are synthesized those nutrients for the growth of bacteria, and this bacteria start colonizations in that junction of root as well as shoots, and creates the disease known as a crown gall disease. So this is a natural phenomena of agrobacterium infections to the plants. But what happened in the laboratory conditions or manipulating conditions, this tDNA has been modified. What happened? This tumor-inducing gene has been removed out, so it's known as a disarmed plasmid, and we can introduce our gene of interest. You can see in this case, here that foreign DNA has been introduced along with the selection marker kanamycin, and these cassettes have been going to be transferred into this TI plasmid. And this TI plasmid, when artificially infected to the plant cells in tissue culture conditions, this transfer that gene, that foreign DNA, as well as kanamycin resistance gene. What happened? This kanamycin resistant gene provide protection to the plants or growth to the plants. Those are containing this particular gene, and rest of the plants are not able to survive. Those are not having this particular gene of interest. So in this case, you will be selected plants, those are containing only this particular cassettes or gene cassettes. So based on that, you will visually observe, and subsequently again you can go to perform some molecular analysis and then biochemical or agronomical performance to see the your trace specificity in that transgenic plants. So this is in the laboratory conditions that you can do by agrobacterium mediated transformation methods. But when you are generating those plants in India, specifically considered transgenics, there are certain regulatory bodies are available. So if you are working on any recombinant technology, first you have to get the permission from Institute Biosafety Committee. That is governed by your own institute. Those are research institutes or universities or colleges wherever you are working on any recombinant DNA technology. Then first you have to get permission from IBSC. This IBSC is going to be informed to RCGM, that is known as a Review Committee on Genetic Manipulations, and it is governed by the DBT, Department of Biotechnology, Government of India. And subsequently, this RCGM transfer to the information to GEAC, that is known as a Genetic Engineering Appraisal Committee, and it is directly governed by the Climate Change Ministry. So what happened? If you are going to perform your experiments in open environmental conditions, then finally you have to get the permissions or approval from the GEAC. So these are the regulatory authorities in India. Those are governed by Government of India. In case of world, you can see several crops have been already been commercialized by using genetic engineering or transgenic approach. Nearly 41 countries in the world and 22 crops have been developed by transgenic approach, and those are commercially growing in the different part of the world. And most of those crops are basically considered cotton, maize, and soybean. So you can see that it's not very new. You can say several countries have already been growing these transgenic crops for commercial cultivations. And there is a one very famous example of BT brinjal that is commercially growing in the Bangladesh. You can see that insect known as a eggplant fruit and shoot borer. It is completely infect and destroy the fruits of brinjal, and you can see you are not able to purchase this kind of infected fruits or brinjal from the market. And this is a responsible, this particular insect is responsible 30 to 60% yield loss, and even for spraying the frequently by insecticides. So this is the disadvantage of what we can say normal practice by using insecticides, where you are going to be contaminated the environment and also not getting protection against to these insects. What happened? This BT gene has been introduced in that eggplants brinjal in India in 2010. They tried to commercialize, but somehow due to some what we can say issues or technical or what we can say that regulatory issues, government of India was not allowed to grow those kind of BT brinjal in India. But somehow that Bangladesh is very pro about this kind of technology, and their farmers and their government were very kind and agreed to get this technology from India that was developed by Myco company. And initially 20 farmers in 2014 have been started this technology and growing BT brinjal in their country. And subsequently 2018 data have been shown has been significantly increased, where nearly 27,000 farmers are growing because that farmers are getting benefits of this technology. And here is the statistics. It has been shown that the non-BT brinjal has been given that income nearly 357 USD per hectare. While those farmers are growing BT brinjal, they are getting that incomes nearly 2,150 USD per hectare. So the profit they are getting more than sixfold. So this is the advantages of this technology. And also they have reduced the use of pesticides, that is nearly 60%, 61% reductions of pesticides use. So they are also protecting their environment. So this is the success story of BT Brinjal. And second one is the golden rice. I think you heard about this. Golden rice, what they did, they have introduced the beta carotene pathway in the rice seeds by using transgenic approach. What they did, they have developed generation bird in golden rice where they have taken the gene phytin synthase from daffodil, then CRT gene from bacteria, and lycopene beta cyclase gene from again daffodil. Those are the plant derived, and CRT is basically a bacterial derived. And all these three gene combination they have introduced in a targeted manner to the rice seeds, and they have shown that carotenoids pathway started to synthesize carotenoids, and that is containing 1.6 microgram per gram, but it is in very less amount. So what they did, they have improved this technology, and in the next generation golden rice generation 2 have been developed, and in this case they have taken phytin synthase from maize, and they have taken CRT gene from bacteria, but they didn't consider lycopene beta cyclase. So combination of only two genes, one is derived from maize, that is phytin synthase, and second one is derived from bacteria have been taken, and the you can see that carotenoids content has been increased 23-fold as compared to the control. So this golden rice is a can see in the colors because carotenoids has property to give the golden color, and that's why it is known as a golden rice as compared to the normal rice, and it is ends with the beta carotene or precursor of vitamin A. And that the Philippines is going to be commercialized. They have already been approved this golden rice for commercial propagation and cultivations. Now I will come about CRISPR-Cas approach. This CRISPR approach you can know about, you know that if you are going to be, this is the complete set of gene in or genome in the living cells, and when you edit or make that any changes, those might be addition, deletion, or replacement is considered like genome editing. So I have already mentioned chemical mutagenesis and physical mutagenesis. Those are basically a random mutagenesis or random mutations in the genome. So it is not very specific. But in case of CRISPR or genome editing based mutations, you can see it's working in a very precise manner. This is the original genetic material where this is a DNA, it is going to be transcribed into the RNA and subsequently form the amino acids or protein. That is the end product. But if you make the changes in the genome in precise manner where you have deleted this guanine nucleotides, what happened is subsequently you will see that there is a changes in the friendship mutations, or there is a changes in that particular because this nucleotide has been deleted. So there is a changes in the sequence of DNA, and subsequently you can see messenger RNA is formed, but protein is not formed because there is a stop codon have been generated as well as there is a friendship mutations. So what happened that end protein will not form. So you can able to disrupt the function of a particular gene in a very precise manner. This is the beauty of this technology as compared to the traditional chemical or physical mutagenesis. So there are different methods of that genome editing like ZFN, TALENS, and CRISPR. And CRISPR is most commonly utilized. And how it works, you can see this is derived from the prokaryotic system or bacterial systems. What happened in the when that in natural conditions, this prokaryote cells is able to get protection against to the virus infection. This virus is transferred their genetic material, that is basically DNA. This DNA is going to be integrated in the CRISPR locus, and it is transcribed to form a pre-CRISPR. And this pre-CRISPR RNA basically recognized by that Cas protein. And this Cas protein has endonuclease activity. When the same kind of virus is going to be infect again to the prokaryotes, they may make some that what we can say complementation. They recognize this complementation stand, and this Cas protein has endonuclease activity that can chop it out the function this particular virus DNA, so that this virus is not going to be infect to the prokaryotic systems. So this is the innate immunity of prokaryotic cells that has been used to develop that what we can say genome editing tools. And you know that two lady scientists, Emmanuel Carpentier as well as Jennifer Doudna, got Nobel Prize of this precise mutations using genome editing in the field of chemistry in 2020. So why that regulations are required for genome editing? There are three questions come in our mind. What if the genome editing goes wrong? What is there's undesirable effects, and why? What if it can't stop? Considering these questions, the government of India has decided to make the guidelines, and in this guidelines they have divided that CRISPR approach in three categories: SDN1, SDN2, and SDN3. In SDN1, basically considered based on that generation of knockout events. There you can implement this approach to silence the function of genes. So you are not going to introduce new genes, but you are silencing the function of a certain genes in the genome of a living organism. Second one is basically SDN2. In this case, you are going to be introducing a small portion of that foreign genes in the genome. So in this basis of that, the government of India considered SDN1 and SDN2 technology, especially in case of plants, will not be followed strict regulatory guidelines that we usually followed for transgenics. So they are considered SDN1, SDN2 as a deregulated and exempted for the strict regulations. Only IBSC is able to approve if you are showing that there is a no introduction of new transgenics in that particular crop plants. So this is that advantages, and several countries have already been approved and deregulated these kind of SDN1, SDN2, and they are not following any transgenic regulations if that crop plants have been generated by genome editing, especially SDN1 and SDN2 approaches. There are certain examples of that. Those have already been commercialized, like you know that genome edited tomato that has been known as a GABA tomato, where that gamma aminobutyric acid has been improved, and this GABA amino acid is responsible for the relaxation and low blood pressure. So this is the first product in the world that has been commercialized by Japan. The second one is the polyphenol oxidase in that mushroom has been silenced, and this particular enzyme is responsible for the browning feature. So if that gene has been silenced, that anti-browning feature has been introduced, so that you can see that white हेलो।
हाँ सर, आ गया फिर।
अभी अभी ठीक है ना?
हाँ, अभी ठीक।
या
अभी
मैं
ठीक।
फिर से स्टार्ट करता हूँ फिर, है ना?
हाँ सर, उस स्लाइड से सर शुरू कीजिए।
या वहीं से करता हूँ मैं। Now, is it okay?
Yes sir, it's okay.
Yeah. So in case of like genome editing, as I am saying that they had so many what we can say applications where you can implement to develop abiotic stress tolerance, biotic stress tolerance. You can enhance the micronutrients. You can develop the seedless crop and also reduce the anti-nutrients. So, but it has challenges also. First is that you have to be know the whole genome sequence of a particular crops so that you will get less off-target effects by designing the guide RNA in your approach. Then you have to be optimize the protocol of tissue culture to develop or transfer that particular traits, and also the selection of delivery mechanism, like which kind of methods like agrobacterial mediated transformation, particle bombardment, or PEG, which kind of delivery methods you would like to use, that you have to be optimized in case of genome editing approach. So this is that pipelines you have to know. First is the selection of gene. There are the softwares are available that can help you to design the guide RNA. And this guide RNA you can go to transfer into the CRISPR vector. And after cloning, you have to be know that which kind of delivery methods like RNP based, then what we can say agrobacterial mediated transformation, particle bombardment, or nanoparticle based method according to optimizations of the process you will use. And after that performance of the transformation, you have to regenerate the plants by using tissue culture approach. And after generation of the plants, you can go to screen by different sequencing methods or bioinformatic tools. So this is the pipelines in case of using genomatic approach. So there is a huge market of genomatic. You can see it was started in the last 10 years only. And in this green color bar has been shown that it's a significant increase or capturing in the market as compared to the transgenics or conventional breeding approach. So it is a huge or potential future of this technology if somebody is starting working in this direction. So in summary, CRISPR-based approach offers a great potential towards the crop improvement. CRISPR-based genome editing products are a step ahead and safer due to the precision, and SDN1 and SDN2 category of genome editing are exempted from the regulations in India, and we are expecting very soon those will be available in the market. Now I am just covering about the second parts where I have used this technology or approaches for the biofortification of banana. I have started work in 2012 in biofortification of banana, where we have collaboration to Australian University known as the Queensland University of Technology. There is a Dr. James Riddle is our collaborators, and in this picture you can see our Prime Minister Narendra Modi has been visited over there in 2014, and Prime Minister Office of India is taking so much interest of this particular project, and every 6 months to 8 months we have to be submit the reports about the progress of this project. And secondly, about that exploratory work where we have designed and developed our own technology by funding from the indigenous supports like DBT as well as NABI core grant. And in this project, we have different collaborators like BARG, TNAU, HR, and RCP. Those are the different institutes in India, and they are working on different approaches. While NABI is the coordinator of this project, as well as we are working on provitamine enhancement. So why we have taken that banana in our choice crop? Because it's a tropical fruit crops cultivated in more than 130 countries. India is the largest producer of banana and contributing nearly 26% of the world banana productions. The banana fruit is available throughout the years at the reasonable price. And this is the most choicest and suitable food crops for all in all age group pupils like infants, adults, and old people. But why we have to work banana by using genetic engineering approach? Because most of the cultivated varieties of banana are replied in the nature. So they are genetically sterile. So breeding is not possible in case of banana. So that's why we have to be dependent on the genetic engineering approaches, that is by transgenics or genomating. So that's why we have chosen banana for the nutritional enhancement. Then vitamin A deficiency is one of the major problem in the world that you can see, especially developing countries, including India, have been reported a severe vitamin A deficiency. And it is reported more than 6.5 lakh children die per year just because they are not getting sufficient vitamin A in their diet. And another more than 3.5 lakh children go permanently blind. So vitamin A deficiency is responsible for mortality, blindness, night blindness, impaired immune system development, and impaired brain development. In case of India, it is reported nearly 57% of the children, those are having less than 5 years age, are considered severe vitamin A deficient. And it is reported more than 3 lakh children die per year just because they are not getting sufficient vitamin A in their diet. How you will get vitamin A? Vitamin A you can get from two major sources. One is the animal, second one is the plants. In animal, you can get in the form of retinol or vitamin A, that is a direct source. But in case of plants, you can get a precursor of vitamin A known as a pigmented carotenoids, or also known as a provitamin A. So alpha carotene, beta carotene, and gamma carotene are going to be converted into the vitamin A in our body. But beta carotene has highest activity to convert into the vitamin A. So why we have to work on plant sources? Because approximately 375 million people worldwide, and around 30% of the Indian population is vegetarian. So that's why we have chosen banana or plants as a source for the production of vitamin A. How much vitamin A we have to be increase in this statistics? You can see that we need 1,000 microgram vitamin A in our diet per day. That is known as a RD or recommended dietary allowance. The conversion ratio of beta carotene to vitamin A is 1:6. So if you would like to get 100% RDA, that is 1,000 microgram for vitamin A, you have to eat 6,000 microgram beta carotene per day. So when we skin the Indian banana germplasm, we find out that single banana is usually contain 400 to 500 microgram of beta carotene. So if we would like to enhance the 100% RDA or 100% required of vitamin A from single banana, we have to be increase nearly 15-fold high content of beta carotene, that is a 6,000 microgram. So that is the estimation, so that we can get 100% RDA from single banana, we have to be increased nearly 15-fold high content of beta carotene. So this is the carotenoids biosynthesis pathway. Plants, you can see that all that carotenoids resources comes into the carotenoids pathway by the activity of one gene, one enzyme known as the DXS, deoxyxylose 5-phosphate synthase. This provides substrate flux in the carotenoids pathway, and subsequently phytine synthase is going to be converted that gynenyl diphosphated into the lycopene by subsequent enzymatic steps, and this lycopene is diverted into the two directions. One is the synthesis of lutein or alpha carotene by the activity of lycopene epsilon cyclase. And second one is that lycopene beta cyclase is converted lycopene into the beta carotene. So what we did, we have used the mixture of two techniques like overexpression, gain of function, where phytine synthase and DXS gene was used for the overexpression, so that substrate flux can be go to synthesize beta carotene, that is our target molecules. And loss of function CRISPR-Cas approach, where we disrupt the function of this gene lycopene epsilon cyclase, BCH and CCDs, because lycopene epsilon cyclase is diverting the lycopene other side. So if we can silence or disrupt the function of this gene, what happened that all resources can go to synthesize beta carotene. And BCH and CCDs are responsible for the degradation of beta carotene. So we had silenced those function of this gene so that beta carotene will be stable. And PDS we have used as a proof of concept. So because it is responsible for the formation of chlorophyll. If you have silenced this gene, you will see the albino phenotypes. So proof of concept we have demonstrated by using CRISPR approach in banana by silencing of PDS. So we have optimized the protocol of tissue culture. As I mentioned, this is the one of the most important requirements for implementing any genomating tools or any transgenic or any genetic engineering approaches. So you can see we have very efficient protocol of embryogenic cell suspension culture formation. So many embryos have been formed in liquid media, And this embryos have been regenerated in the tissue culture media under that aseptic conditions, and subsequently those were acclimatized and developed in the full plants. So this is the capacity where you can see we have generated thousands of plants from the 1 ml packed shell volumes of embryogenic cell suspension culture. This is the steps that we usually followed in our laboratory. First one is in silico analysis, that is a bioinformatics or software-based analysis, where we have to be just identified the gene and predict the function of that particular genes, and then subsequently characterize the function in the bacterial complementation systems or in the aeronosis as a model plant system. After characterization of the function of gene, we perform transformation in using our optimized protocol, and after transformation by using tissue culture approaches, we generated the plants and under that in vitro conditions, and subsequently plants acclimatized in the soil pots and transferred into the greenhouses. And after that, plants were transferred into the net house, because we have to be followed the biosafety regulations, because we are working on genetic engineering approach, and government of India has mentioned that if you are working on any recombinant technology, you have to follow their regulations, especially growing those plants under the containment or control environmental conditions. And after harvesting the fruits, we had performed the HPLC to see that carotenoids content in the fruits. So this is the approach that we had followed. We had also published several research papers in this direction. If somebody would like to go in details, they can see these papers. So that number of plants have been generated and acclimatized in the greenhouse. You can see then fruiting was started in this net houses, and then we had performed their analysis. So first one is the genome editing, where we had edited PDS gene. This I mentioned is responsible for the chlorophyll biosynthesis, while lycopene epsilon cyclase, BCH, and CCDs we had considered for the genome editing to stabilize as well as enhance the content of beta carotene. Those work have been published in different journals. Here you can see PDS was published in Functional and Integrative Genomics, lycopene epsilon cyclase editing was published in Metabolic Engineering, and CCD editing was published in Journal of Experimental Botany. These are some of the review articles. If somebody would like to go in details, how that genome editing is helpful to improve the nutritional traits or any desirable traits, they can go in details about these publications. Then, as I mentioned, PDS is responsible for the synthesis of chlorophyll. You can see over here, when you silence or edit the function, then chlorophyll synthesis have been stopped. And also, you will not get the carotenoids. So in this case, what we did, we have designed the construct for CRISPR editing to silence the PDS gene. And in tissue culture stage itself, you can see that albino phenotypes have been generated because chlorophyll synthesis has been blocked. And visually, you can see this kind of observation in edited lines. Similarly, some of the plants have been generated, and you can see the albino phenotypes. So visually, you can identify it by the editing of PDS that your approach is working. And when we had performed sequencing of these edited lines, we have seen that some stop codons have been generated, and due to that, protein is not able to synthesize PDS. And this PDS was not synthesized, so that it blocked the synthesis of chlorophyll as well as carotenoids content in the edited lines. As compared to the control, that control, you can see that there is a sufficient amount of chlorophyll has been synthesized, but edited lines, there's a reduction or completely depletion of that particular chlorophyll biosynthesis. Similarly, in case of carotenoids, also you can see that control have been sufficient amount of carotenoids biosynthesis, but edited lines have not been synthesized. So this is the first report in the world that have been shown editing is possible in case of banana, and that we have demonstrated in 2018. Then our second target is lycopene epsilon cyclase. As I mentioned, it is responsible for the diverting lycopene into the other pathway site, so that when we edit this gene, that beta carotene synthesis has been increased. This was the hypothesis. Based on that, we have designed the CRISPR construct. We had performed the tissue culture to generate the plants or edited lines, and you can see that again we had performed the sequencing to see that there is an addition or deletions, and there are different assays that have been confirmed to see that there is editing or not. Along with sequencing, we have performed those assays, and after performing HPLC in the fruit, we have seen that certain lines have been shown high depositions of beta carotene, and it is also visualized the dark yellow color. So we have noted nearly 6-fold high content of beta carotene increased when lycopene epsilon cyclase gene was edited in banana. And then again, we had performed some agromorphological analysis in our net houses growing plants on comparable features to the control. That work was published in Metabolic Engineering, and it was the first report in banana to improve any nutritional traits. Then our second target is CCD. As I mentioned, it is responsible for the beta carotene degradation. I have identified two contrasting cultivar, Rasthali and Nenden, and noted that CCD genes like 4, 1, and 8A are present in abundance amount of the fruit pulp of these contrasting cultivars. What we did, we have taken these three genes and performed their expression in different fruit developmental stages. That is started from 3 weeks to the fruit ripening and fruit green and fruit ripening stages. And similarly, in the other varieties, Nanten, those are containing high amount of beta carotene also considered in the similar expression for these CCDs. Also, we have estimated carotenoids developmental stages of fruits, and you can see that in this variety, Nandan naturally contain the high amount of beta carotene. But this variety is commonly grown in the southern part of India, Kerala only, but not the other parts. So what we would like to understand, this Rasthali variety is containing very low amount of beta carotene. The expression of CCD4 is very high at the early stage of fruit development. So this CCD is responsible for the degradation of beta carotene. And we have made that correlations and find out that CCD4 is the potential candidate for beta carotene degradation in case of Rasthali. Then we have characterized the CCD's function by using GCMS and find out that CCD4 is responsible for the degradation of beta carotene as well as 10-apo beta carotenol, because this CCD4 enzyme is particularly functions or disrupt or just targeted this beta carotene and apo beta carotenol and form that byproduct known as a beta-iodine, but not for other carotenoids. So it makes sure that CCD4 can be used for the gene editing in case of Rasthali, so that function of the gene can be disturbed and it will not be synthesized that CCD4. So subsequently, carotenoids content will be increased in case of Rasthali. We had performed the transformation protoplast of Rasthali, and also we had generated several plants by using non-transgenic approach. There's no basically integration of the Cas9 that we have confirmed by PCR approaches. Edited lines have been generated, and subsequently we had performed that their metabolic analysis in leaf and root tissues, and it have been shown that the content of beta carotene has been increased 2 to 3-fold in these edited lines. That work was published in Journal of Experimental Botany, and it shows that transgene-free editing is possible in case of banana. And fruits have been already reached in the fruiting stage, and that we are going to be analyzed very soon. And second approach is the overexpression. As I mentioned, DXS provide the substrate flux in the carotenoids pathway. So what we did, we have cloned this DXS from the variety of Nantan, and DXS2 is expressing very high amount in the fruit pulp of Nantan. So that gene have been taken and cloned in the plant expression vector with the fruit specific promoters, so that expression can be happen only in the fruits. And you can see that after some plants were grown in that our net houses, and those are looking in the completely normal and comparable to the control. When we cut those fruits, it is completely turned to the golden color, because that all carotenoids flux have been transferred from upper pathway to down pathway of carotenoids biosynthesis. And that might be the reason after overexpression of DXS2, we have seen that golden banana, those are showing nearly 20 to 30-fold high content of beta carotene as compared to the control. This is the normal banana that we can see in the market. So these are some other pictures of golden banana, where you can clearly show that content has been increased significantly as compared to the control. Then we had performed some bioavailability assays, so that we have to make sure that whatever beta carotene we have increased in the banana, it is basically converting into the vitamin A. So for that, there are some assays are available in that animal cell lines known as the caco cell lines, and we had performed that cellular uptake assays, and we find out that single banana is provide 100% RDA, that is a 1000 microgram for vitamin A. It is basically converting beta carotene into the vitamin A from the single banana, is providing sufficient RDA requirements of vitamin A for human. And now that project is in the second phase, where we are performing multi-location event selection trials in the open environmental conditions by following all DBT biosafety guidelines. As you can see that we have got the permission of GEAC in 2020-22, because now we are performing our trials in open environmental conditions in these 5 locations: Tirchi, then Coimbatore, then Gujarat, then Assam, as well as Mohali, Punjab. So 5 locations trial have already been started. And you can see this is the summary of our journey that has been started from lab to land for golden banana. The work was started in 2013-14, where we have optimized the protocol of regeneration or tissue culture. Then next 2 years, we optimized the protocol of transformation and their regeneration. And subsequently, you can see plants have been generated and transferred into the net house. And 2019-20, we have developed that nutritionally enriched banana. Those are showing the golden color in the net house conditions. Now we are performing event selection trials in open environmental conditions, and plants are now going to be commercialized in 3 to 4 years if things goes fine and government will approve. So this is the reports published in Times of India and Indian leading newspaper, where it is clearly mentioning that after mustard, that banana, rubber, and maize on the way that can be released in the environmental conditions in India. These are the current trial status in different places of India. You can see Nabii in Gujarat, that Tamil Nadu, as well as in Assam. So we are growing our plants in different environmental conditions in India. So we have to see the impact of that environmental conditions and agronomical performance of banana or our edited or transgenic lines. And here you can see this is just a slides. It is showing that normal banana, if you would like to get 100% idea, then you have to eat a number of fruits. But in our case of golden banana, a single banana is sufficient to provide 100% RDA. That is a layman language that people can understand that how this technology is superior to provide nutrition by using biofortification in banana. And this is that government of India is supporting us a lot. When that Science and Technology Minister Dr. Jitendra Singh visited in our institute, he have appreciated the efforts by developing this golden banana, and that government of India is fully supporting us so that this product has to be reached in the market. And this is that another video that I have given to interview one of the renowned science communicator, Dr. Mr. Pallab Bagla. If somebody would like to go in details about how we have developed this banana from lab to land, you can go and see this particular interview. And I'm really thankful to my lab colleagues who have contributed a lot for the development of this technology. And I acknowledged to our project partners, Australia, NRCB, BARC, TNU, IHR, NAU, AU, and our research team at NABI, our executive directors, as well as funding agencies, BIREC and DBT for their support. So thank you so much. I have given, I think, more time. I have to be finished before 15 minutes. But sorry for this delay, and now the floor is open for questions and answers.
No, sir, it's a perfect. No, we are on time, and all are admired about your slides because it's very easy to understand from various levels of our participants. I think students are very happy
Yeah, thank
for
you
your
so much.
yes, for your
I
slides.
try to make my slides so that I think so.
Yes, yes, and thanks
When the
for
students of a school going, they can also
yes.
understand.
Yes,
I try to.
that is the purpose that we ask people to register, so we can and get an idea that which people are watching and slides can be made in that way. So now we are going to start, Lily. Now we are going. Now we are going to start the
Yes.
interview session. Shoma will conduct the interview session. Okay, sir.
Yes.
So,
Meanwhile, you can collect the questions and let's. All right. Hello, sir.
yeah, hi.
First of all, thank you for the absolutely wonderful presentation. So our viewers, you are now about to, we are about to now start the speaker interview session. This session promises you to give insights into our speakers' life, their scientific journey, challenges during their PhD and or research work, views on food security, on GM crops, upcoming field of research in plant sciences, collaboration projects, manuscript writings, and much more. So, sir, we will start this session with a little background information about you. Could you tell us a little bit about your scientific journey, how you became interested in plant sciences, and what inspired you to pursue a career in crop biofortification and nutritional security?
Yeah, Dr. Soma, it's a very interesting questions because I belongs to village in MP, Madhya Pradesh. I started my schooling in Sohagpur. that is basically one of the village place in Shangabad district that is known as now in Narmada Sambhag. So Narmadapuram, sorry. So in that place, basically, we don't have any English medium school at that time. I started schooling in a Hindi medium school.
Mhm.
So subsequently, usually I have visited nearby places where the plants are growing, especially farmer communities are. You can say there are so many farmers are there at that time. So I have very curious to see that how plants are growing, how they are surviving, how they are producing the seeds, and how they are going to produce fruits. So I was curious at that time how it is happening in the plants, and they have certain life cycles, and they are going to be again the same features in the next generation. It developed some interest in my thoughts that how it has happened and how we can improve the yield as well as quality. So at that time, I started thinking about this direction, and I was lucky because I got that guidance and directions of my senior colleagues as well as my family. They supported me a lot and they guided me so that I started journey from there. And after MSc that I completed from Bhopal, at the Barkatullah University, and I got that lucky because my senior was doing PhD at that time in Lucknow,
Mhm.
and he told me that you can come over there. And when I went over there, I was fortunate to meet my supervisor, Dr. Rakesh Tuli, was the senior scientist at that time in NBRI, that is a
Mhm.
National Botanical Research Institute, and he encouraged me a lot to work in this direction, especially how plants can be used to produce vaccine, as well as how we can develop the uh, genetically improved variety like insect resistance variety of groundnut.
Mhm. Okay.
So he guided me, and after that, I started developing my interest, and that journey has was started during my schooling time. And still, I am curious to learn new things in this direction.
What were some of the major challenges that you faced during your PhD or in your research career, and how did you overcome them?
Actually, failure is the part of any kind of success story, we can say. I
Mhm.
also got so many failures during my PhD as well as during my this what we can say after joining as a scientist. But during PhD, what happened? After 3 years of laborious efforts to optimize the protocol of tissue culture, regeneration, transformation, groundnut, uh, after 3 years, I got good success, but somehow that time I was ill due to some physical problems. I got operation for hernia, and I went to my native place for 2 to 3 months. And that time I lost all my transgenic plants. Those have been developed after 3 years efforts. Nobody was care at that time because some limitations. So I was very much upset and very down at that time because all that efforts I put in last 3 years was basically destroyed. So, but I was the luckiest one because I have very good friends. They motivate me. They
Mhm.
encouraged me, and my supervisor is very renowned scientist. He also encouraged me and supported me a lot.
Mhm.
So I started from zero again, and after 3 to 4 years, I have so many good successes, as well as I have published my all chapters, all thesis chapters, in different journals.
Yes.
So I think failure is the part of any success story, but depends upon you how you have taken those kind of failures in your life.
What do you think are the key factors contributing to food security? And how can plant sciences and crop biofortification address global food security challenges?
Yeah, it's a very interesting questions and completely linked with my current research objectives.
Mhm.
During my presentation, also I highlighted that population is increasing day by day, and especially in case of India, we are the largest populated country. I think we should not feel proud of this particular achievement because we have so many responsibilities now. We have to provide sufficient food, sufficient nutritious food to our populations. How we can do this? Because we have limited land, limited resources. So breeding, as I mentioned, it has certain limitations. You can't improve that all crop plants by using plant breeding approach. Now that genetic engineering comes in the picture, and we are very fortunate, like genome editing approaches or techniques are available now in that scientific community, where you can use and improve the traits. So for specifically biofortification, I personally think genome editing is not only the sufficient approach, but we have to be believe on transgenic or genetic engineering, other genetic engineering approaches also. So in case of banana, you can see we have developed this nutritious enhanced variety by using transgenic as well as genome editing. And we are hoping that it will be commercialized because our neighboring countries like Bangladesh, they have already been growing that BT brinjal for that their consumptions. And it is a I'm not 100% sure, but it is very high expectations that BT brinjal is also growing in India because Bangladesh and India border is very close.
Yes.
So most this is a very high possibility that we are already consuming those kind of genetic engineering crops in our food chain. So I think that we have to be believe this technology and utilized it for the human well-being.
So, talking about crop improvement, what are your thoughts specifically on genetically modified crops being a solution for enhancing nutritional content and addressing food security concerns?
Yeah. As I mentioned, it is already linked with that our population, that growing populations. How you can provide nutritious food without implementation or utilizing this kind of technology. But transgenics are playing major role, and I mentioned in one of my slides that nearly 22 countries have already been growing these transgenic crops in their soil. And those are basically utilizing for consumptions of humans as well as animals. Those things are already in our food chain, and we didn't notice any kind of negative impact for the environment as well as human beings. So I think government of India should also be encouraged these kind of technologies, and they are definitely supporting. That's why we are working. So we are hoping very best that in future those technologies will be available in the market, and we all can be utilized.
In your opinion, what are some of the emerging or upcoming areas of research in plant sciences that hold promise for addressing nutritional security? And are there any innovative technologies or approaches that you are particularly excited about in the field of crop biofortification?
I particularly what we can say that is seeing that emerging technology is AI, artificial intelligence. It is implementing it anywhere or everywhere.
Yes.
So, and also for crop biofortification, it can be definitely utilized where we can be a predict or design a particular what we can say trait or what we can say the gene or protein. Those are important for particular nutritions. And after all, designing or predictions by using AI tools, we can implement practically in that technology in crop plants. And nowadays we have that genome editing because government of India is also supporting these kind of technologies. Not in India, even other countries also they are supporting, and it's a very promising technology, a very evolving technology. So I personally think combination of AI as well as genome editing will be the game changer in future.
So, talking about working and in different countries, research everywhere, how do you approach collaboration with researchers from different disciplines and from different institutes?
This is very interesting questions, and I personally believe that a single person can't do everything. So always you have to have very good collaborators. In case of mine, I am very fortunate and lucky that because I have very big team. In India, I have nearly 8 collaborators, different institutes, this banana project. And every collaborator knows their responsibility very well. They are working on different aspects because they have different expertise. Like some of them have very good expertise on agronomical characteristics of plants, and they are good for that nutritional analysis. And in my institute also itself, one of the scientists who is my collaborator, Dr. Mahendra Bishnoi, is very good in the nutrition science, and he is supporting me a lot about all that nutrition aspects of that golden banana. So definitely collaboration is very, very important.
So, what advice would you give to young researchers about writing and publishing scientific manuscripts in plant science journals?
Uh, First, I think they have to be develop the habit of reading. When you start reading that good articles, research articles, review articles, you automatically develop that what we can say that art to write a manuscript, because you will evolve your thought, your process, your writing skills. So most important is you have to be read more and more specifically about your field, what is happening worldwide, and that review article, what is the emerging tools and techniques and approach that renowned scientists are thinking. So review article definitely will help for these directions. So I think so reading is very, very important.
All right. So, based on your experience, what are some key skills or qualities that young researchers should develop to succeed in the field of crop biofortification and plant sciences?
I realized if you should be very focused about your approach. If you are diverting, you would like to do many things, it is not possible. First, you have to be have very focused about your approach and do hard work. I personally realized there's no substitute of hard work. Maybe you are very intelligent, you are very sharp to understand the things, but if you are not very hardworking, especially in research field, you will not get that much success. So you should be very hardworking in this.
All right, then. So we are at the end of our program here, and now we will go back to Shovo to see if he has any questions that he would like to ask on behalf of our viewers. So, Shovo, let's get back to you.
Yes.
Okay.
Yes, I'm coming. I'm just collecting.
Yeah.
Most of the questions are based of GM
Mhm.
safety and security. But yes, your whole talk is self-explanatory. It means people are maybe joining little bit delay. That's why they are asking the questions. One questions I found in the YouTube that during the biofortifications of one nutritions, is there it has any effect to lower down the others nutrition which is previously available on that crop?
It's a very, very interesting question. As I mentioned, if you are working on transgenic approach, then there is the possibility of integration of gene in the location that can be disturb the function of other important genes. So in this case, you have to be generate a number of plants, number of lines, so that desirable lines, those have no negative impact or no what we can say undesirable effects. You have to be identified. And regarding that, seeing the changes in the nutrition profile of other nutritions, then we go for performing that proximate analysis. That is one of the types of analysis where you can compare the different nutritions in your transgenics or genome edited lines with the non-transgenics or control lines. So based on that, you will identify, okay, there should not be any changes on that particular nutrition. So it's a part of basically screening. We can't completely rule out that there may not be any impact. There is the possibility of impact of other nutritions, but there are ways to just sort out or just identify what is happening in the plants. And based on that, you can select lines or plants, those are comparable to the control with only that your desirable traits.
Mm.
So this is the way.
And another questions is related to the life cycle of 45 biofortified banana crops and control. Is there any delay of fruit development or ripening? Is there any changes?
No, we didn't notice any kind of this kind of impacts in our Mohali net house experiments. But certainly now we are performing open trials in different places in India. So our collaborators will let us know that what is happening about these life cycles, yield, or other agronomical parameters. So I
Mm.
think next 6 to 8 months we will have some good data, and based on that we can say what is happening in our that genetically engineered plant of banana.
Okay. One question is from my personal side that uh, I just wanted to know about your fundings. Means BIRAC, you have a BIRAC funding, you have a others funding. So this work is challenging because of the, as you know, the rules and multiple scrutiny. But also you need fund for those overcome these challenges. You need a lab, polyhouse, net house, and everything. So, can you give some idea about us that if someone going to develop a transgenic plants, which is like your, then where from he or she started and how approach the funding one by one, so ultimately it can go for the lab to fill.
Yeah, it's a very challenging as well as what we can say interesting question. If you are starting your journey on working on transgenic, I think first you have to be design a project. Those should have very promising, and it is clearly reflect that outcome will be sure after ending of this project. So maybe in India, usually you will get supports for 3 to 4 years. And after 3 to 4 years, if you will show that any good success or outcome of that particular project, then subsequently you can again apply for another funding from different agencies. I am fortunate because I that project for banana biofortification was supported by BIREC and Government of India, specifically our Prime Minister office is taking direct interest
Hmm.
because we have collaboration with Australia where we got some technology and then subsequently we developed it in our own. But for that other researchers, I can suggest first they have to be right project for 3 to 4 years. It
Hmm.
should have very strong base because I realized now that getting funding is very competitive. It's not
Hmm.
very easy to get support in the project. So first you have to be designed very good and smart project, and based on that, if you get supports for 3 to 4 years, and based on the success, definitely you will get subsequent funding. So that is
Yes.
the
So,
way.
one by one connection, one connected to
Yeah,
others in that way.
exactly.
And yes, in our last webinar, um, PK Gupta sir is here, and he also mentioned that India forced to commercialize BT cotton because already farmers are started to growing it in different areas of
It's a
India.
BT brinjal.
Yes, now it's the BT brinjal. No, he told about his BT cotton in
Cotton.
2002.
Acha. Okay,
So
okay.
I also didn't know the story, but he told us that government forced to start accepting the BT cotton because already it's gone in it. But yeah, definitely we are in Kolkata, so many
Yeah,
very close
yeah.
to Bangladesh, and we are also hoping that we are consume the BT brinjal. But yes, and personally, my research interest and story is a little bit similar to you. Like I did
Mhm.
my PhD in BT jute. I
Oh,
expressed Cry1 ABSC gene in jute. And
okay.
then right now my project is metabolic engineering of jute lignin modifications.
Okay, okay. Very nice.
So
Great.
it's and this year, one mini project has done on planty bodies productions. So
Oh,
it's somehow when I go through your talk, somehow I recollect that okay, one day, what, it's
definitely, it's an opportunity to make some collaborative work
yes,
because
yes,
we have
and
same interest in the same
yes,
direction.
and
So yeah.
also I have in jute, very
Mhm.
few people are working. So I also
Yeah,
struggle.
yeah, yeah.
Sometimes I struggle a lot during some simple things like which is very common in others plant, but not in the jute.
True.
Now, during my related work and ongoing work, I have a question on the PDS.
Hmm.
Because it is a chlorophyll, it's related to chlorophylls and biosynthesis pathway also, and also
Right.
I now I realize that okay, it's also related to vitamin A,
Yeah.
provitamin A pathways.
Hmm.
So those plant has low PDS, means
Hmm.
they are their leaf is not green because they are a little bit yellow or light green. So,
Right.
is there any effect of their leaf structures? And obviously, there's a vitamin A is a good is another thing, but due to the low chlorophyll content in the leaf, due to the mutations in PDS, is there any photosynthetic related issues of those plant you have observed, or is it at par?
No, no, you will not get photosynthesis because PDS, as I mentioned, and you also know about, it's responsible for chlorophyll biosynthesis.
Hmm.
And in the downstream, it is going to be synthesized carotenoids.
Hmm.
If you silence or disturb the function of that gene, you will not get chlorophyll biosynthesis, as well as you will see that no depositions of carotenoids.
Okay.
So due to that, we have seen so many kind of abnormalities, like plants are not able to survive because there is no chlorophyll synthesis. Plants will not be survived for a long time. And
Hmm.
also that height of plants, growth of plants, we have seen some abnormalities because sometimes we can see variegated phenotypes.
Okay. Hmm.
Like in case of banana, it's a triploid, three
Hmm.
alleles of each gene. So
Hmm.
what happens sometimes that all three alleles will not be edited.
Hmm.
So it will be considered like heterozygous editing. You
Hmm.
will get some variegated phenotype. There is some green as well patches and that yellow color of leaves.
Hmm.
So
Hmm.
plants are going to survive, but what we can say, those are not very healthy.
Okay.
So
Hmm.
this kind of observation we noted in our that
Hmm.
study, and also we have highlighted in our publications
Okay.
how that you can see this kind of observation.
Hmm. Hmm. And just I wanted to share my experience in PDS. Sometimes mutations of PDS is very challenging. Means getting the albino plant sometimes is very challenging for jute. I think I don't know for your case, but yes, we got. I'm not yet to have any albino, but yes, difference in chlorophylls I have found.
Okay, okay, okay.
So have you
I,
seen
I ha
such
one
type of?
one yeah, one
Hmm.
question about that your observation. Have you performed sequencing also to make sure that there's a mutation or not?
Yes, I perform, but now I have to check the because I prefer the exon one. I
Okay,
prefer to mutant in exon one because
yeah,
logically it's the first, so I stopped the first.
exactly.
But I think I need to check the whole because it has 14 exon in.
Uh,
So maybe
okay.
others will give the effect, but right now but I thought that don't waste more time on that, so I
Yeah,
just
yeah.
I make the manuscript ready in
Yeah, yeah, that's fine.
in that way. But
It's a, just
yes,
a proof of concept, so you should be
yes,
focused on the target, not
proof
for
of
this. Yeah.
proof of concept like thing. But yes, sometime getting the actual mutant is for the PDS. I'm sure for the target gene it will be easy, but for PDS maybe I don't know, but maybe for the jute it may be challenging.
Yeah, yeah, it depends upon the crop also, and which
Yeah.
kind of guide RNA you have designed.
Yes,
Nowadays,
yes.
people are
Oh,
going for that multiplexing also. That
yes. Hmm.
target.
And that and I'm also planning in that way that multiplex or targeted multiples in case of jute. And people are still I'm sure the recording is available so you can go through their comments. They are very happy about your
Okay,
presentation because
okay.
what I can understand that they are very connected to it with their
I was
syllabus.
thinking I was thinking it might be boring because I have
No, no,
taken
no,
only
no,
one and a
no.
half hours.
No, yes, yes. Our session
So,
is generally 2 hours. So many
okay.
people are coming at the end also. And those are
Okay.
coming the end. Sometimes they miss the link when we finished it
Okay.
early.
Okay.
Like upcoming two webinars, first and 15 is a former professor from Canada. So this
Okay.
will be the 1 hour webinar. And many people
Okay.
will
Okay.
miss that webinar in that.
Okay. Okay.
Okay, sir. So it's we are going to end because now
Yeah.
the topics are health related concern about GMO and all. But yes, all answer is available. Means these are the
Yeah.
Yes,
We can
and
search on
we
that.
are the scientists, so it is our responsibility to check everything. Then
Yeah,
we can go for the field and yes out. So we are also very serious about what we are going
that's
to
true. That's true.
going to propose or give to the society or farmers. So yes, we also take care of this thing. And
Exactly.
thanks is a great pleasure to
Thank you. Thank you so much,
find
Dr.
you.
Majumdar. Thanks a
I,
lot,
I,
Dr. Soma.
yes,
It was really nice interactions, and we see that in future we can meet personally in some
yes,
future.
I definitely, and for
Yeah.
for all the audience also, if you
Yeah,
want to
yeah. All are most
collaborate
welcome.
with our
Yeah.
with us and with our speakers. Today is Siddharth sir, so he is also open because few peoples are from Africa. They are asking for they have a wild type of potatoes, and they are wanted to use in biofortification projects. So formulating a project is very difficult. So that's why I'm asked them to collaborate, write a mail to the Dr.
Certainly,
Tiwari
certainly.
sir. So for the more planning on that.
Well,
But yes,
exactly. That all the audience are most welcome. They can directly contact me through
hmm,
email or just they can write Twitter or like Facebook, whatever comfortable they feel.
hmm,
And I will try my best to give their answer their queries, as well as if some of are living in India and they would like to come to visit Navi, they are really most welcome.
hmm,
They can see this amazing facility. They will interact to other scientists also, and I am hoping that they will learn more about this. And
hmm.
in future also, I will advertise two PhD positions in my group. If
Okay.
somebody is really interested on genome editing, especially in banana, or developing different tools, they are most welcome to interact with me.
Obviously, and when you publish this type of advertisement, please forward me. We have a Telegram group actually
Oh,
where
very
I
nice.
posted many things which can't be posted in openly, like many
Okay, okay.
papers, many study materials, books, and
Yes. Okay.
these groups. These people are also communicate each other through this group. So
Oh, very nice.
sometimes people are asking for the full paper, and someone give it to them. So
Personally, yeah.
I'm just formulated this group, but this is the self-sufficient group. So many times I posted those are other than Bioingene programs like any
Mm-hmm.
advertisement, job, postdoc positions, and all. So I can forward these things in
Sure,
that.
sure. I will share with you. Definitely, I will share with you.
In January 2024, I'm in Navi that time, but I did not able to find time for the visiting your greenhouse and skin house. But definitely in
Yeah,
next
yeah. Next time
time,
you can
yes,
please join us. Yeah,
yes,
yeah.
I'll ask you to visit and real thing. And
Sure, sure.
let's see. I'm hopeful for Jute also. I'm doing some research. I'm confident on that. That maybe someday, sometime. Let's see, sir.
Sure,
Okay.
sure.
Thanks.
Definitely.
And
Yeah.
for the all the participants, I'm going to giving the certificate application form which should be submitted to within 5 hours, and those are registered will get the certificate. It can be downloaded from our website homepage after 4 5 days. And our next webinar is fast at 6:30 Indian Standard Time on 1st May. So see you in the next webinar. And sir, thanks for accepting our invitations and a great talk. It was a great
Thank you.
talk, and
Thank you so
it
much.
will be in recording in YouTube. So maybe many students and many young peoples are got the interested. They can start from DNA to CRISPR DNA. So it's a whole story, and I'm glad that you brought your plant. antibodies work here, because I'm not sure that I'm getting any speaker on antibodies in India, but I'm glad something we also focus that okay, antibodies can be,
Exactly.
and
Exactly.
obviously different techniques are available, but it need to be checked, and yes, when it is applicable, then we should use them to get the more benefited. uh,
Sure.
thanks, and see you soon, sir, and
Thank you
definitely
so much.
we'll collaborate for something.
Sure, sure, sure. Most welcome. Sure.
Thanks,
Thank you
sir.
so much. Thanks to all of that audience. And thank you so much.
Thanks.
Bye.
Bye, sir. Okay.